Hello, my name is Cynthia, and I'm a CalTeach or Math and Science Education student at UC Berkeley. During the 2022 summer, I had the opportunity to intern at the Joint Genome Institute, or JGI, and learn about the web resource PhycoCosm. Today, I will be going over a quick tutorial on how to use some of the tools available on PhycoCosm. Before we start, here is a quick overview of what I will be going over in today's video. First, I will give a quick introduction of the JGI and PhycoCosm. Then I will discuss endosymbiotic gene transfers and conduct a case study on that using PhycoCosm's tools. So a quick introduction. The JGI was established by the US Department of Energy in 1997. The goal of JGI is to advance genomics for clean energy and environmental cleanup. It is a national user facility that sequences genomes and makes genomic information available to thousands of users worldwide. PhycoCosm is a tool developed by JGI. It hosts a large collection of algal genomes and tools for analyzing them. This analysis helps us better understand algae, which is important because they are a major agent of photosynthesis and are currently being looked at as a target for many different biotechnologies, such as biofuels or bioenergies. They also play an important role in carbon cycling. For my research, I'm looking at how genes move around an algal cell. Algae are photosynthetic, and their ability to perform photosynthesis comes from an evolutionary event called endosymbiosis, where many millions of years ago, a non-photosynthetic eukaryotic cell took up a cyanobacterium, which can perform photosynthesis. Over time, the two became host and symbiont, with the cyanobacterium losing genes to become the plastid that we now know as a chloroplast. During this process, some of the genes from the plastid got moved to the host nucleus, and we call this process endosymbiotic gene transfer, or EGT. It's interesting to study these EGT events because they give us insight on how the nuclear genome reacts to plastid genes coming in and which genes we choose to accept. So now let's head over to the PhycoCosm website. On the home page of PhycoCosm, there is a navigation tool that takes the form of a tree of all the different groups of algae represented on PhycoCosm. Currently, there's 159 different genomes hosted on PhycoCosm. I can click into a group at an internal node here or at the end of the branches here. Alternatively, I can use the search bar or click on this drop-down menu to select the group I want to look at. Clicking on the names here will take me to that algae's group portal. To start my analysis, I will use PhycoCosm to look at the organisms in the chlorophyta or green algae group, which is over here. Here you can see a more detailed list of the chlorophyta genomes available on PhycoCosm. Clicking over to tree along the top lets me see the different species grouped by evolutionary relationship. But in chlorophyta, there are many different classes of organisms. The tree is a dynamic tool, and clicking on the nodes lets you collapse or expand groups like this. So the species I want to look at today are Micromonas, which is over here, and Chlamydomonas, which is over here. And these two are in different groups within chlorophyta. So first, let's take a look at Chlamydomonas, or clammy. Clicking on the name of the organism takes me to its genome portal. Clammy is a model organism and the best-known alga. This is the homepage for the genome portal. It includes a general overview of the species, some interesting facts, and where the genome was retrieved from. In this case, Phytosome, another JGI portal focused on plants, for which clammy is a unicellular model. There are many other tools that I can use to look at clammy along the top here. First, let's take a look at the plastid genome by clicking on the Organelle tab and selecting chloroplast from the drop-down menu. So here you can see a diagram that shows all of the genes on the plastid genome color-coded based on what kind of protein they code for. And there's a key for that over here. This table has more information about the plastid genome of clammy, such as assembly links and the number of protein coding genes. I'm going to pull up the plastid genome of Micromonas to compare to the clammy one, and that is over here. So you can see that the Micromonas plastid has fewer protein coding genes. One of the genes that is on the clammy plastid but not the Micromonas one is YCF4, which is over here. YCF4 is a protein involved in the assembly of photosystem 1, which is needed for photosynthesis. If this gene is not in the Micromonas plastid, then how is that algae still able to perform photosynthesis? An EGT event may have occurred and the gene may have been moved to the nuclear genome. Let's explore to see if the YCF4 gene is in the nuclear genome of Micromonas. So first, let's download the gene information from clammy, where I know it is present in the plastid genome. So the download tab is over here. From the download page, you can access different CLAMI files. So I'm going to click on expand all, and you can see all of the available files. There's files for the genome, its genes, and its proteins. This is a file I'm interested in. This is a plastid protein file, and I'm going to unzip this file, look for the YCF4 protein, and then look for its amino acid sequence. So I've retrieved the amino acid sequence for YCF4 here. Now I'm going to take this sequence and look for it in the Micromonas nuclear genome using the basic local alignment search tool, or BLAST. To get there, I'm going to go to the Micromonas genome portal and click on the BLAST tab. So I'm going to go ahead and do that now. So now I'm back on the Micromonas genome portal, and now I'm going to select the BLAST tab. Now I'm on this page, I'm going to paste in the amino acid sequence. Since I have the amino acid sequence and I want to compare it to the nucleotide sequence to look for the presence of a gene, I'm going to use tBLASTn. I want to look at 3 different databases: ESTs, or express sequence tags, which is essentially RNA, filtered model transcripts, and assembly. RNA shows that the gene is expressed, filtered model transcripts show that the gene is annotated, and assembly shows where the gene is in the nuclear genome. So once I have these selected, I'm going to go ahead and run the program. So these are the 7 matches for the amino acid sequence I put in the BLAST search. The RNA results here show that this gene gets made into RNA. Assembly scaffolds show the location of the gene, and you can see that this gene is located on chromosome 8 in the Micromonas nuclear genome. Gene catalog shows that there is an annotation for this gene, meaning that this section of DNA is indeed a gene. Clicking on the assembly scaffold result, I get taken to the browser view, where I can see this section of the Micromonas nuclear genome and its features. Here, I can zoom in or out to look at the surrounding genes. So the green lines down here are the RNA alignment to the DNA, meaning that this gene gets made into RNA. So I'm going to click on Gene Catalog, which takes me to the protein page for the protein made by this gene. So this is the protein page for the YCF4 gene. At the top of the page is a protein ID, which is a number that refers to this specific protein. Here I can also see the number of exons and a description. This over here is a gene ontology, which predicts the function of the gene. This is predicted to be a photosystem 1 YCF4, which is what I'm looking for. I can further confirm this by looking at the information from the scientist who named this gene, an outside biologist using PhycoCosm. So it looks like this scientist also named this gene YCF4 and added some additional notes about its function. Under that, it says that there are 2 transmembrane domains, or TMDs, predicted, meaning that this protein is predicted to get embedded into a cellular membrane. I can click onto that to take a look. So the red regions over here are where the TMDs are predicted to occur within the protein sequence. And now I'm going to head back to the protein page. This section here shows the gene structure. I'm going to click onto that for more information. The red is a protein coding sequence, and the blue is an untranslated region, which is not translated into the protein but may still be important for gene regulation. The gray shows the nucleotides upstream and downstream of this gene. So below this is information about the amino acid sequence. Everything down here is aligned with this bar over here. The dark blue is a signal peptide, which means that this protein gets secreted and moved to a specific place inside or outside the cell, such as an organelle. In the case of an EQT, we will want the signal peptide to target this protein to the plastid. The yellow boxes show the protein domains, and you can see their names over here. The green shows the amino acid with exon-intron splicing boundaries. This protein has only 1 exon, so it is a continuous line. The gray bars here show other BLAST results of organisms which have YCF4. So the more similar proteins are higher up, and as you scroll down, the organisms get less related. The first 2 hits are YCF4s in the nuclear genome of other green algae closely related to Micromonas. But down here, the protein is encoded in the plastid of a species that is not related to Micromonas. Clicking on the gray bar takes you to their alignment, and clicking on the name takes you to the amino acid sequence. So to wrap up, today we used PhycoCosm to do a case study where we identified an endosymbiotic gene transfer event. This information can be used to help confirm evolutionary relationships and gain insight on how the nuclear genome decides what pieces of plastid DNA to accept. So we identified a problem where the YCF4 photosynthesis gene in the clammy plastid genome was not found in the Micromonas plastid genome. And then we hypothesized that this gene is now in the Micromonas nuclear genome as a result of an EGT event. And we confirmed this by using PhycoCosm's tools. So first, we use a tree tab as a dynamic tool for visualizing the evolutionary relationships. Then we use the organelle tab to identify an interesting gene. Then the download tab to get to the amino acid sequence of the protein that gene encodes. The gene we look at today, YCF4, is in the chloroplast of clammy, so we downloaded it from that portal. The gene is absent in the chloroplast of Micromonas, but instead found in its nuclear genome. To confirm that it is in the nuclear genome, we switch over from clammy to the Micromonas portal, then use the BLAST tool to find the YCF4 gene in the Micromonas nuclear genome. Once they found the gene, we click onto it to get to the protein page to learn about the YCF4. Here we learn about what the function of the YCF4 is, its protein domains, and what other organisms make a similar protein. We also saw that this gene had a signal peptide that transports the protein made by the gene to a plastid, where it functions. So this process can be used to analyze other genes to identify similar EQT events. This is only a small subset of PhycoCosm's capabilities, and further exploring this web resource will reveal many other tools useful for looking at algae. So here are the links that we use today. This is a link to the PhycoCosm homepage. This is the Chlorophyta group portal. This is the Clammy genome portal, and this is the Micromonas genome portal. Thank you for taking the time to watch this video tutorial.